So we are joined by exhibiting artists from the Oceans 4.0 exhibition. We'll first invite Marco Barotti, followed by Robertina Šebjanič and Gjino Šutić, as well as Dr. Rachel Jeffries, to speak about some of their research and projects. Each of the three artists will discuss their projects currently exhibited in the Oceans 4.0 exhibition, which monitor and reflect and imagine the future of our threatened oceans. In nature, clams are detectors of pollutants. They serve as tiny filtration systems. Inspired by this natural phenomenon, Marco Barotti presents his new work, Clams, a kinetic sound installation triggered by water quality. Live data is streamed by a sensor placed into the ocean and converted into an audio signal. Barotti's clam sculptures, made from recycled industrial plastics waste, are triggered by this signal, generating an evolving soundscape, creating an experience that allows us to see and hear water quality in real time. So I'd like to invite Marco to the front and just give him a big round of applause.
I thought that it would be an introduction to my practice. So I brought also a few other projects. If you don't mind, I introduce you to my, also all the other works, which are at least two of them. And then I will go to the clams, and then we can talk about it. And then I think it's easier to go on with question and answer right away. My name is Marco Barotti. I'm a media artist, and I'm based in Berlin. I studied drums at the Academy of Jazz in Siena. This is my background as a musician. And in 2008, when I moved to Berlin, I had the great opportunity to collaborate with many artists from different disciplines, from architects to choreographers and designers. These people inspired me and challenged me to look at my creativity from another point of view. So I shift my music path and career into a maker somehow. And this is what I became. So my personal love feelings for nature and animals became a practice. What I did in 2016, to start a little bit from the beginning, during a residency in New York, I experiment with visualizing sound. So just, attaching brutally different materials to the membrane of loudspeaker. I experiment on how this material would react when you shoot in bass frequencies, so infrasound. I show you a short video. This series is called New York series, of course. It comes from the place where I was. And you can see somehow the beginning of it. So with the outcome of this residency and this experiment, I began creating a series of works that merge audio technology, consumer objects, and waste into kinetic sound sculptures. These sculptures, they deal with environmental issues and play with their resemblance to animals. This is the first one of its kind, the swans. These installations are mainly presented in urban and natural environments. So as I said before, I'm going to show you 2 or 3 works. The swans, first. Swans are satellite dishes swimming into water. The work reflects on tech waste, the way we consume technology, and is somehow raising the question of where this technology ending up once we are done with it. The soundscape played by the swans is a composition made with people breathing into brass instruments and a layer of bass frequencies that generates natural compressions and metaphorically imitates the sound of real swans. So the work has also been presented indoor. This is the Kunsthalle Osnabrück. Indoor, the swans, they swim in a pool of 7 meters with the water painted in black, and they reflect the architecture that surrounds it. So this one was inside the church. What you see on the right side it's supposed to be an underwater motor that wasn't really underwater on this version. It creates a water stream that destroys the reflection and it gives lives to the swans. When the motor stops, The reflection slowly comes back and the swan stands still. And this is a loop that goes. I go to the next work, the woodpeckers. The project aims to open a discussion with the citizens about electromagnetic pollution. I designed small robotic birds inspired by real woodpeckers that are able to transform electromagnetic pollution in real time into audible and visible drumming patterns. They use urban metal objects as their instruments, and they create a rhythmical composition and constant transformation using the data, the invisible data that surrounds us. The sonification that happens in real time. I use this EMF meter that translates the data into an audio signal. Then the audio signal is sent to Ableton Live, where it's low passed into bass frequencies, and then it's sent to the woodpeckers. The head of the woodpeckers is a loudspeaker, so it functions as the first video that I showed you. The bass frequencies, they generate a push and pull movement that somehow recreates the data in its dynamic speed and amplitude. This is a video that I shot in different cities in Europe.
Awesome.
The project, of course, exists in nature. This is Terschelling, an island in the Netherlands, where the woodpeckers were actually pioneered last year. And I would like to go to the stars of the day. And the reason why I'm here, thank you very much for having me, by the way. The clams are about water and plastic pollution. So as Mark was saying before, why clams? Because in nature they are a detector of pollutants, and they serve as tiny filtration systems within our ecosystem. Scientists in the States, in Florida, they are deploying these little creatures in rivers and pick them up months later. And by the colors of the tissue, they are able to define what kind of chemicals or heavy metals are in the water. In general, every year, an estimated 8 million metric tons of plastic enter our oceans. This plastic waste decompose into microplastic, releasing toxic chemicals into the seawater, severely damaging marine life and entering our food chain. So I decided to create sculptures that look like clams. They are made out of recycled plastic and they are triggered by the water quality. Now, how does this work? There is a sensor that measures water quality that can be placed anywhere, in the near of the installation or in the other side of the planet. This sensor streams real-time data to a chain of computer applications that translates the data into an audio signal. The audio signal is then sent to the clams, and it generates the live evolving soundscape as well as the opening and closing movement of the sculptures. What you see here, the preview that you see here, it's a much smaller version of what I usually present. These are 8. Usually I do 80, so it's a much larger scale. The sound is traveling through the installation into 8 different audio channels. So it's a sort of surround sound that is somehow emphasizing the real data coming in. You will hear as well later maybe the changes that you hear in pitch and amplitude are basically when the parameters they go up, the pitch goes up and the clams they move faster, and when the parameters they go down, the clams move slower and the pitch is going down. The plastic that I use is coming from industrial waste. They are mainly pharmaceutical bottles, and it's recycled for the consumer packaging market. So it's PETG that is actually illegal in Europe, illegal in Canada. The process takes quite some time. It's quite DIY. So this is a machine actually to print T-shirt. These are the micrograins which are coming from the cleanup of the plastic. So I do transparent and blue transparent, and then I melt these grains together. Only this process is about 10-12 minutes. Then they need to cool down, and you need to take care when you take the parts off, you don't destroy the plastic, otherwise you need to restart from the beginning. So imagine this for 80 clams, it means 160 times. And I have 2 sets of clams, so it means 320 times. So what I'm doing here then, I use a vacuum former, and I use real shells as molds. Shells, they work as trees, so from the circle they have in their surface, you can deduct how old they are. So what I'm doing, I'm printing the history of the animal into the recycled plastic. And then you can see they're all cut by hand and done by hand. So it's an infinite process where it becomes meditative, being in the atelier and spend hours after hours after hours doing this. They're attached together with magnets, and there are cables running under the sand. There is 2 molds. One is coming from Hong Kong, one is coming from Russia. There are 2 travels, I think 2 of the cities that inspired me the most for the project, so I decided to print these two shells. Thank you.
How do the oceans feel our impact? Robertina Šebjanič and Gjino Šutić aquaforensic illuminates invisible pollutants, residues of human consumption and monsters in our waters. Documenting the artist's in vitro experiments, the project shows microorganisms dying in weak solutions of pharmaceuticals, 20,000 times weaker than the average human dose. Interrogating the negative impact we are having on our water habitats and life forms, aquaforensic questions our solidarity and empathy with water systems beyond our human perception. I'd like to invite the 2 speakers to the front and give them a round of applause as they do so.
Thank you for a lovely introduction. I'm Robertina Šebjanič.
I'm Gjino Šutić. We will present the project Aquaforensic, as mentioned.
Because with Aquaforensic, it was like our project of last year we have been doing research during the residency at Ars Electronica, which is like a really amazing center for arts, science, technology in Linz. But the project was also kind of a result of our, because me and Gjino, we know each other for many years. We wanted to collaborate, but we wanted to collaborate on something which we really both really feel passionate about it. So before we will speak about the project more in depth, which is also presented here at the exhibition of Oceans 4.0, we will just give you a brief of intro of who we are outside of this joint venture. So I will start to speak with how I even came to work with the aquatic environments and why I started to do that. Because I actually started to work with marine environments during my residency stay in Izmir in Turkey in 2011. And there I was actually coming across lots of jellies because there was like big jellyfish blooms in the Izmir Bay. So I was there actually to do something else, but then because this was present, this was there, and this is also, it happened not in the time it should happen. It was like February, so these kind of jellyfish blooms are happening much later in the year. So it started to be telling us also, you know, like to be reflecting to these kind of bioindicators, if we could call it also like that. And on the other hand, my interest into jellyfish was actually because of their amazing regenerative powers also, especially the collagen, which is like which is also part, we have it also in our skin, but they have it much more and it's much more present. So I will not speak too long about this because, you know, it was like actually 5 years ongoing research which went quite in depth into understanding the species by itself, understanding why we do use it as model organism in biology, especially these ones, the moon jellyfish, which I work with. And they have been opening many discussions. I'm happy to speak about this later on if somebody will be very curious about. But just to point out, the project actually resulted in 2 variations. In first variations was more like a relationship between the jellyfish and the robotic machine. And this was kind of showing if the jellyfish are rolling and they're running it, then the robot was actually the puppet, which was like controlled by them. Very simple and the quickest explanation. And in the second point of the project, because, you know, I just couldn't, you know, in the first one, I really wanted to be just like this more technical in one sense. But then I saw to bring this kind of empathic relationship also with something which is so foreign to us and so different because we can't relate to it, you know, it doesn't have the same eyes, it doesn't have this big eyes, you know, which lots of like pandas or other animals which we like to refer to and we feel more comfortable with them. But, you know, they're a bit different, but they have this kind of mimesis, this kind of movement is constantly happening, you know, the pulse. And this is something which we can all relate. I think you as a drummer, you will agree with me, you know, there is this rhythm which is very important for us humans. Like it feels comfortable because it's our inner rhythm also, somehow. So during my research, I started to do lots of kind of like sonifications and trying to understand what is happening on the sonic side, and we didn't know it. So part of the project is actually a result of audiovisual performance, where me and jellyfish, we are performing the sound together. So partly it's controlled by them, partly by me. On the end of the performance, I leave, and I like to explain the project actually with the words of Paul Celan. There is still songs to sing beyond mankind. And what I try to bring out is actually the understanding that, you know, the way how we understand the language, maybe the other species, they have something completely else. They do still communicate, but it could be on chemical levels, it can be on some different receptors. So this is like brief about this project. And then in last years, my practice started to be much more related towards the marine biology, and I spent lots of time in different marine stations or next to the seas, or I have this privilege to be colleague with Gjino, who has amazing lab in Dubrovnik also. So I started to do lots of underwater recordings also, like recordings with hydrophones. So it's kind of like passive listening to the environment. And on the beginning, it started with this kind of like being bored and listen, and then I started to notice that most of my recordings, even if I managed to record some sounds of the nature, it was always having also us humans present next to it. So it was like lots of boats, lots of technology which is present. And on the beginning, I was kind of deleting that out, but then I realized that I'm trying to do the Photoshop of the nature in the sense of like presenting it different and having the idea of nature which we always like to romanticize. So I was going like back and forth in my ideas what to do with that. And then I decided to do the compositions with it. Kind of compositions which they combining like bioacoustics. This means like the animals, the sounds, and also their communication, how they talk with each other, because in aquatic environments, the sound is very important sensor, much more than the visual. Like I like to say humans, we are visual animals, but in the waters this changes, you know, because it's like 200 meters down, and very quickly your visibility is like losing, so you can't see so well anymore. So the sound starts to be very important, and we use this as humans also a lot in the traveling in the oceans and seas, but with this we are disturbing so deeply and very strongly the animals. And this was kind of like one of the starting points how we started to speak about it, because this is one of these kind of pollutants which is invisible in the sense of like we do not notice it. Till you don't put the hydrophone in and listen with your own ears, you can explain, I can explain to people this many times, but I see the effect when I have workshops. In the moment we hear that, starts to have like something else, you know, because it's kind of immersiveness which brings us to that. The last project which I would explain about my practice is like from this year, I was working together with a big team from Institute, Mutant Institute for Mental Narratives in Madrid, Matadero. They just opened this institute this year, and they tried to bring also the awareness of this kind of like ecological situations, which is happening with the world which we are living. And I think that people, all of us, start to be more and more aware of that something should change. And maybe it's us, but also it's like how to change this in bigger numbers. So they invited me to develop a workshop for 500 participants, which first I was like puzzled, because mostly I do workshops for 10 people. So it was a bit other challenge, I would say, but with good team you are able to do that. So it's of course, you know, these kind of complex projects I'm never alone. There is always like group of people which we are working together. And what we developed was actually 40 points in the Manzanares River. We have been working with the river of Madrid. This was kind of the commission had this insight, you know, so they invited me to work something with Madrid River, whatever, it was like connecting with the workshop. So what I developed is kind of like water monitoring workshop, which is not bringing only the measurements to the people, but because we have been ambitious, we also publish a booklet, which is like very thick fanzine. We have been joking that we kind of try to compete to have like the thickest fanzine in the world. And in this fanzine, we also wrote about texts about environmental justice, about meditation with the river. So it was like different variations of how to enter to the river, not just like the river, because, you know, the river, when you say river, mostly you think about the water. But the river is also the riverbed, the ecology about it, the animals which they are part of it, and everything which is connecting with it. So we have been working on that also. So one of the results of the group action was also this infographics, which is showing the situation and from all the measurements, because of course we did also the pH measurements, everything what you have to do to see the quality of the water. But then also what was important for me at least was also to have this kind of different strategies, how to be more impactful to people. And I would say that also, Marco, like you said before, I also try to, I try to bring this kind of soft activistic approach in the sense of like, let's rethink together the situations where we are and how to solve this further. So that's from my side.
Okay, so I have like a related but different approach. I come with more official scientific background, even though I'm mostly self-taught scientist and an artist. I'm running Citizens Research Institute, Universal Research Institute in Croatia, Dubrovnik and Zagreb, with facilities in both cities, doing research in science-related art, pure science, and production of related events. I've been producing lots of programs since it opened in 2013. Maybe best known of these programs is Biennale Summer School of Science and Art that I do host in Dubrovnik. Also, there is like Bioelectronic Academy, educational program for youth in bioelectronics, combining technology, biology, and engineering skills. The biggest program I did was Black Clinic Triennale Festival of Biological Art and Technobiology, because that's primarily my media. I experiment, I'm specialized into bioart and technobiological installations, using mostly living organisms, their tissues or cells as the media to create innovations. And this innovative process of creation, for me, I see it as a combination of science art, because for me there is no difference between two if you are creating something new. So this is the examples of one of my first works, growing human replacement organs from scratch in my own kitchen using bacteria from environment, which is not very useful. It actually spoils wine into vinegar, but I use it to synthesize 3D scaffold that can be mixed with stem cells to grow human replacement hearts from scratch. It can be used also for other organs. I played with this material for a while. So the other science art innovation that was produced during Art Incubator, Sound Art Incubator in Zagreb, is microbiological speakers, microphones, and drums, where I use the bacteria to cultivate nanocellulose and process it in a specific way to produce membranes for speakers, microphones, and drums. And by doing so, like to grow technology from scratch for cheap, to come up with more eco-friendly, efficient solutions for creating technology. One of my maybe scientific, like most complex installations so far I did was the Moment of Death for Touch Me Festival in Zagreb, where I played with forensics and reversed forensics for a while. So the topic of the festival is combination of science and art and human senses. So this was like commissioned piece. For them, I played, I built do-it-yourself environmental probe equipped with billions sensors and put the corpse of a pig in a grave site in soil, connected it with this probe, so it collected all possible environmental data you can collect digitally. The data was digitalized and sent online via internet, streamed real time to the gallery, where it was recreated back into physical world in this black chamber, where the audience could step in and have like 5-minute experience, sensory experience of being in a grave. So it synthesized the smell, the humidity, the temperature, the texture, like everything was there, streaming of the audio and the video. And the idea was like to do a reverse of sensory deprivation, to temper into all possible senses humans use, so they can't distance themselves from the experience. And by doing so, to force people to cope with the idea of death that we are usually trying to avoid, to see what happens. So it was like psychological experiment also in parallel, and it was accompanied by a reverse forensics workshop, how to get rid of a corpse at home. But the idea behind this project was also to be like scientifically innovative, because when you do like forensic experiments in body farms where you check like something on the corpse, we usually use like pig corpses as a models. Always like one aspect is checked, and the idea here was like to collect all of this raw data to maybe collect something that you haven't considered while designing the experiments that can be analyzed afterwards. I also worked a lot with ecology, ecological engineering, and so on. And this is actually how Robertina and I met and started collaboration. In 2016, I did this piece like Sushi Roulette, do-it-yourself analysis for the presence of microplastics at home in your own kitchen, and accompanied exhibition. And this project actually is a step up of the project from 2014-15 that we did together in Bergen, Norway, where we started finally, after knowing each other for a few years, where we finally started like proper collaboration in exploring invisible water pollutants. Here we focused on analyzing noise pollution in waters and do-it-yourself analysis of microplastics in algae. And we flirted with the idea, we were thrilled with this, thrilled and scared at the same time with this aspect of invisible pollutants and their personal effect on each one of us. And we found that this topic is interesting for general audience because if like plastic pollution, we all know about it, but most people don't care about like the whole continent made of plastic floating somewhere in Pacific Ocean. But the idea of you eating plastic in your own plate, that's something that makes us thinking about pollution in general. So in this project, Aquaforensic, we went further. We explored a lot the topic to see like what hasn't been explored in the invisible pollutant area. Okay, and we wanted to analyze the seas and waters because there was like lots of mythology related with that, like monsters in the waters and so on. And we always saw the animals in waters as monsters, but are they really? Or the monsters are something new that we can't see? And for us, that was these like pharmaceutical pollutants.
And we also to continue this story about the monsters and how to rethink this. It's like also like what is the context of the aquaforming, you know? Not farming, you know, like farming with so on, but you know, to relate to terraforming as an idea how humans we are changing everything. We started to rethink also that aquaforming in the sense of like this what is coming from us through the sewage to the dark waters, through the rivers, back to the oceans and seas and so on in the whole cycle. It started to be our kind of main idea which we wanted to research. So this we applied to the residency, which partly was executed in, we shouldn't be together too much, I think. So it partly was executed in Ars Electronica in Linz, and the second part in Ur-Institute in Dubrovnik, so which actually gave us good perspectives, because in Linz we have been working on field research on the river in the Donau. So we did some sampling and see what is happening in the context of this there. And then also with similar what we did in Ur-Institute in Dubrovnik. Our focus was mostly on these like chemical pollutants, which they are mostly antibiotics and different pharmaceuticals, which we are all actually using it on everyday basis. And what we like to say is we are using and abusing it. And we never say that like I'm actually having also some health issues, I have to take some particular pills every day, you know, and so on. And it's just like the thing is that our bodies are very, very wasteful in this sense, because, you know, when you take the pills in, only maybe max 20% of the pill stays actually inside and it goes into the body circulation. 80% goes out, you know, and going out, this means it going into the toilet. And dark waters are these kind of sewage waters, which they are bringing all this somewhere. So maybe you did hear something about, you know, the rivers full of cocaine or, you know, illegal drugs, but it's the same thing also happening in huge quantities on legal drugs. And what is the thing is actually it's also, okay, partly it's also from us humans, and this was the basis of the research which we have been doing it. But the big pollutants are actually the companies and the industrial farming, you know, in the sense of with all the antibiotics and everything which goes from the farming for the terrestrial kind of animals, but then also aqua farming. Like with monocultures, we are changing everything. And with this kind of monocultural approach in the agriculture, there was like too much of this happening. So. But that's like something which we would like to continue further in the research also, because last year, during 2 months, we decided to be really focused.
So we focused on like beginning and the end of this like food chain in waters, on microorganisms, unicellular algae, phytoplankton, cyanobacteria. because like the life cycle in water starts with unicellular algae that feed everything and ends with bacteria that decompose everything. So if there is like some change in some effect, negative effect on these microorganisms, the whole ecosystem is affected. And we also, as Robertina mentioned, focused on consumable drugs. So the one that we all have at home. So we actually, the residency, as mentioned, took place like in two localities, in Linz, Ars Electronica, and in Dubrovnik University Institute. In Linz, we actually focused on developing the theory, developing the experiments, and developing the tools for research. So the first thing we did was like making do-it-yourself drone for underwater sample collection that can go like far away that we can't, on the places we can't reach. In Linz, we tested the theory that actually pharmaceuticals are released into waters by collecting the water samples from different localities, then first screening them for the presence of fecal bacteria to see if there is like wastewaters being released there, like human waste. When we found the localities with these waters, we took more samples and analyzed them for the presence of consumable drugs, illegal and legal drugs, to check out if there is some inside. Of course, we found them. We did this also like in Dubrovnik. We found that different cities prefer different drugs. But yeah, so this was like to prove the theory. Then we collected the sea bottom samples to extract like different microorganisms, mostly unicellular bacteria, unicellular algae, cyanobacteria, and protozoa that we exposed to nanoscale quantities of the human consumable pharmaceuticals, the approximate equivalent to those we found in waters, to check out what's happening. We expected some negative effect of the drugs on like microorganisms, for example, that the human tranquilizers will make them more peaceful or something like that, but we actually didn't expect that the effect would be so strong. And actually this is the video of the first experiment we did, exposing protozoa to smallest possible quantity of ibuprofen, painkiller that all of us use at home, to check out what's happening. So if you could see, it became like peaceful for a while, then it started to run away from the source of the pollution. Then it became disoriented. Now it's trying to expel the pollutant from its own body and dying in the process. We did like a million of these experiments. So we took also like 2 interns and provided them with learning hands-on experience during the summer in Dubrovnik.
We have been trying to be also efficient in the sense of like get the data as much as possible in the concrete timing also to see what is the results, because, you know, it was, as you mentioned, Gjino, like on the beginning we have been really expecting more this kind of more behavior changes, but not so much that there will be diluting in this kind of quantities. And then during the process of like how to actually speak about something like that, you know, like how to put now this kind of research, which is like very based on microorganisms, which most general public and also us, we don't know them if you don't work with them on everyday basis. It's really hard to know what is the change because we don't know it, you know. It's like you don't know, like with the dog you really quickly know if it's hungry or it's happy or it's like whichever, or at least we project that we know that. So when we have been thinking how to represent this in the exhibition context, we decided to build this kind of modular systems, and we show you. So this was like in the Biennale just recently this year in Wrocław. But what we decided is actually to have this kind of, these black ones, they are little models, where you have in the inside of them, there is these little videos happening in constant loop. Because what we decided is to not, you know, we had in vitro experiments, we did that phase, we have the material. We didn't want to do this constantly in real time with people because, you know, I would just like killing more and more, you know, even though this kind of feels like, okay, it's microorganisms, we have been thinking it's better just to present this as document of a death, of a moment of the death, we decided. So it's presented through different variations with this kind of piping systems. This was the bigger version of it. This was the first time we presented it, Ars Electronica. We developed also an artist book, where instead of the artist book, when you look at it, it kind of looks like a pictures book. But in 5 steps, you see what happens to a specific animal, you know. And we just tried to show this. Also, this was in Bourges in France last year. So you see like the organisms, they feel like holograms. And we decided to use this kind of paper ghost hologramic system, which is like one of the oldest tricks. It was used in the theaters in 19th century, and we decided to not have this kind of complex technology to present it, but, you know, to present it to the point of that it's kind of having this feeling of dilutants, and it feels like floaty. So this was kind of what we...
Ghost effect. Yeah.
Exactly, the ghost effect, which we have been looking at. And last year in Bourges, we also have been challenged somehow, but it was excellent to do also a workshop, which we developed then also for representing the project, which we also think is good to have this kind of, you know, when you do this art, science, technology research, sometimes, you know, when you just stay in the one area, like in presenting it in the art context is excellent, but with workshop you can enter more.
Yeah,
So-
I just want to add a sentence about the installation itself. We chose the copper pipes as a framework for the project to illustrate this aquaforming thing that humans do, trying to control the waters, because copper itself is like toxic material that kills all possible microorganisms. So we sense it's proper for this one. And our work group during the research was called like microgenociders because we did so many experiments on microorganisms like to get these results. But it was like thrilling, and in comparison to like purely scientific research, this artistical output is something that everybody can emotionally relate to, which is what we try to accomplish without being too judgmental, because we didn't want to criticize the drugs in general and also the use of drugs because they are necessary, but mostly like misuse and overuse of drugs. And we try to transfer that also to new audiences through this hands-on experience that we prefer actually as a medium, because it provides you with not just like monologue, but a dialogue with people, and this inclusive creative process that also is a learning experience for us. And the art students without any scientific backgrounds adore this, and we also boosted interest in science in them, and it was a cool experience. As mentioned also, we took like 2 interns during the summer to provide them like hands-on experience in work in the biolab. One student from a biology background and one from aquaculture background. They don't do this kind of research in universities in Croatia, so they were thrilled about the experience. So we collaborated with them more on using the aquaforensic residency and all of this collected data as a preliminary research for scientific projects. And there's also like 2 science papers that came out from this. One is soon to be published. This is like the preliminary results that we showed on International Water for All conference, like the influence of consumer antibiotics on unicellular algae and cyanobacteria. And the other one is wider, it's still in the development.
That's a very important side also on the end. I'm sorry, baby, we have been like for 5 minutes too long. It's perfect? Okay, perfect, because we get some signs, so we don't want to go over too much. But it's just like, you know, in these projects, it's always great to have this kind of support from different organizations, and we have been really happy to have this opportunity to show this project, to develop this project inside of Emare EMAP. We have been talking this today already, that to be part of this kind of more bigger, wider network of different organizations gave us also opportunity to show this project in different exhibition spaces, as well as also doing these workshops and so on. But yeah, so we can speak more about it later. Thank you.
Thank you so much.
We're inviting to the front Dr. Rachel Jeffries from the University of Liverpool, who's going to present a presentation titled "You Are What You Eat: How Seals Can Help Us Understand Climate Change." Rachel's presentation will give us a fascinating insight into how scientists from Liverpool are using state-of-the-art stable isotope techniques to understand how climate change is affecting two ice-dependent Arctic predators, the ringed and harp seal.
Hello, thank you very much for inviting me here today. So I'm going to talk about another way that we're affecting our marine planet, and we're moving up to the Arctic now. So this is part of a large NERC, National Environment Research Council, funded programme called the Changing Arctic Ocean. So here we have a map of the Arctic, and it's also become a very interesting place for scientists as well as politically because of the possibility to travel through the Arctic now on some shipping routes. So it's bordered by several different countries, Canada, US, Russia, Greenmark, Denmark, which is part of Denmark, Iceland, and then we have Norway, Sweden and Finland. And so now we have lots of scientists working across these nations and also European nations working with them to try and see what the effect of our changing climate is on the system. So I'll be introducing some of the key players in our project. So this is Piers Buchanan, and he's doing a lot of modelling work. So he's been looking at what's happening to the ice in the Arctic using satellites. And here you can see from 1978 right up to the present day how the ice has been changing. So this is sea ice concentration. And one of the main things that you need to take away from this video is that the ice is really variable between years and also within years. So it varies in extent and thickness within one annual cycle and also between years as well. So this is really important for the organisms that live up in the Arctic. And we go right up to the present day. So you can see how the ice changes between the summer seasons and the winter seasons. So in the video, you can't really see how that's changing between 1980 and 2018, but Piers has put these two different pictures side by side. So here we are in Liverpool, just to orientate yourself, and this is in 1980, and this is last year. And you can really see that the sea ice extent and concentration has dramatically declined over this time period. And it's thought that the Arctic might actually become ice-free in about 25 years' time, which is quite a scary thought. So this is another graph by Zachary Labe in the US. So he's been looking a lot at changes in the Arctic, and he's basically looked at lots of satellite images, and he's just found out that basically it's been decreasing by about 13% per decade. And this is because the Arctic's getting warmer. So the globe is getting warmer, and the Arctic is actually one of the fastest warming places on the planet. So you can see here that air temperature in the Arctic has increased dramatically. So we've gone from sort of mean temperatures of around -1 up to positive temperatures in 2018. And actually last year there were temperatures in the late teens and early 20s, which is unheard of in the Arctic. And so with this, we've seen this shift in sea ice extent, so how much sea ice there is, and in the concentration. So in the Arctic's changing, we've seen forest fires up in the Arctic Circle, which is also unheard of last year and the year before. So ice is really important, not just for the animals that live there. Here you can see a food web. So you can see how the ice provides a platform for seals. Seals use it to haul out on and also to breed on. It's really important for human populations living in the Arctic who rely on this food web, and it's important for some of the smaller organisms. So we were talking about smaller organisms in the last presentation. So things like ice algae, which live, the small plant cells that live in the ice, and then this forms the basis of the food web in the Arctic. But it's really fundamentally important for the planet as well, because the ice actually reflects the sun's heat. And so if we don't have ice, more of the heat from the sun will be absorbed into our oceans. And this is a feedback cycle that's happening. As the air temperatures are becoming warmer and the ice is melting, we're actually getting more of the sun's heat being absorbed by the ocean. So what's going to happen in the Arctic when the ice disappears? So this is some work by some Norwegian colleagues up in Tromsø, and they've been looking at fishery stocks in the Barents Sea, so on the Norwegian side of the Arctic, and they've been looking over the last several decades. And what they found was that in 2004, you can see all the polar fish species here. So we have species that are specifically Arctic species shown in the blue. So this is a snailfish at the top, and then Arctic halibut. And this is where they're found in 2004 in this blue area. And then we have a mixed population. So the population in yellow is a mixed population of Arctic species and also Atlantic species. So we've got Atlantic cod and haddock here, and they are found in this red area. And what we've seen in the last 10 years or so is that actually the Arctic species are being pushed out of their area, and you're getting more of the mixed population and also more of the Atlantic species in the Arctic. And this isn't just happening with the fish, this is happening right down at the base of the food web with the smaller animals. We're seeing what's known as Atlantification of the Arctic, so where we're getting Atlantic species moving up into the Arctic. And this has consequences for some of the charismatic megafauna, or some of the animals that I'm going to talk about next, the seals, which we use as indicators to try and study Arctic food webs. So, for instance, colleagues both in Norway and Sophie, who's in St Andrews, who's also a part of the project that I'm working with, have found that basically in the mid-2000s, the harp seals were doing quite well. They had quite a lot of fat and blubber, and recently, in the last decade or so, this has decreased. And this is to do with the food that they're feeding on. And basically, if you're getting more Atlantic species, they don't have as much fat as the Arctic species, and this means that these seals are not able to lay down these fat reserves. And this is predicted to have impacts on their population. So they use the modelling technique, and they were able to predict that because of this lack of fat in their blubber, we're getting these skinny seals, and basically we're having a 50% decline in pup production. So this is another species that we're looking at. This is the ring seal. So the ring seals really are dependent on the ice and the snow, and they basically build these ice caves in the ice to have their pups. And what Canadian and American colleagues of ours have found, so in the Department of Fisheries and Oceans, is that snow cover in the Arctic is basically decreasing as well. So this is a plot here showing dune snow cover, and you can see since 1970 there's been this downward shift in snow cover in the Arctic. And this has real implications for these seals because they need a lot of snow to be able to build their dens, to have their pups. And so again, using a modelling approach, scientists didn't really think it was important back in the day to look at pups. So they always monitor the adult population, but now what they've done is they've used this modelling approach to see how this is going to affect pups. And what we know is that when there's not enough ice or snow, both for harp seals and ring seals, you might have a female adult that's carrying a pup, and then she might decide to abort, either because she doesn't have enough fat on her to sustain the pup, or because there's not enough ice to be out with the pup at that young stage. And we also see some of these pups are actually being drowned as well because of the lack of ice and snow. So basically the modelling is predicting that as we move forward, we're going to lose more of this population due to pup drowning and miscarriage of young animals. So what are we doing in this project? So I wasn't lucky enough to go up to the Arctic, but my postdoc Camille, she went up a couple of times out on the British Antarctic Survey ship. So this is one of her images going through the pack ice. And she works alongside Camille there. She works alongside James. James has been putting these satellite tags on harp seals to see where they're going, to look to see where they're foraging. And Camille has been looking at the seal diet. Here she is filtering some water. You can see all that lovely green phytoplankton, small plant cells in the water there. So this is our website, and this project's not possible without the funding from NERC, and also without our project partners. We have a lot of international collaborators on this project, and we span seven countries. So it really is a massive team effort. And this is our sampling strategy. So we're trying to use a pan-Arctic view. So we're trying to look all across the Arctic. So we have samples of seals that have been collected by subsistence hunters. So So when they collect the seals, they'll give samples to the scientists in Canada and Norway. And the sites where we have the seal samples from are in pink. And then we have the track data that is it that one? Oh yes. It's okay. It's okay. You can't hear. Oh yeah, for the podcast. Okay, sorry. So we have track data, which is shown by the yellow and green dots here. So this is the seals that James has been able to trace where they're going, where they're foraging. And then we have our cruise data, which is shown in orange, where we've got samples, and additional samples from project partners in blue. So we really have a good coverage of the Arctic. And one of the ways that we're using stable isotopes is that we're looking at teeth. So this is actually a leopard seal, which is found in the Antarctic, but it's a really nice photo to show you the teeth of the seal. So they're not actually, some seals have quite large teeth, but the harp seals we're using have quite small teeth. And basically it's like a tree. So they lay down layers of dentine. Like us, annually we get layers of dentine put down, and we're able to take a subsample of the tooth to do our work. And this is what it looks like in practice. So this is Camille. She's in the archive in Canada. All of those tubes have seal teeth in them. And then this is a seal tooth here, which has been sectioned longitudinally. And Joe up there with Camille in the lab has been sectioning all of these teeth. And we've got teeth back from the 1940s from Norway. So she's sectioned hundreds of teeth, and Camille has analysed hundreds of teeth. So we're able to look at a certain point in a seal's life and try and see what they're feeding on. So we've been doing this using stable isotopes. So carbon and nitrogen are two of the most commonly used isotopes. Carbon is one of the most common elements that we find. And nitrogen is actually really good for looking at food webs, so we can see where an animal sits in the food web, if it's at the bottom or if it's at the top. And we're able to do this by looking at the ratio of these two atoms, which basically have a different structure, and we're able to measure this using a mass spectrometer. So what we tend to find when we're using nitrogen is that as you move from the base of the food web, so looking at these tiny phytoplankton, as you go up the food web, you get an increase in the nitrogen 15 by about 3.5 per mil. So we're able to do some simple maths and figure out where you might sit in the food web. And if you were to measure one of my hairs, say when I was a teenager and I was a vegetarian, you would have been able to tell that I was a vegetarian. Now I eat a bit of meat because I'm married to a Frenchman, so you'd be able to see that I'm a carnivore. So you can use this technique it's really quite a cool technique. And this is some of the data that Camille has produced. So we've gone right back to the 1940s, up to 2000, and basically this is a proxy for where the animal is in the food web. And what we can see is that the seals have basically decreased their position in the food web through time. And that has consequences for, as I've already discussed, pup production and populations. So what's going to happen now? Well, basically we'll use this data, and Camille's also been getting data from another slightly more complicated technique, also based on stable isotopes. And we're going to work with James, who does the tagging, and Sophie, who does some modelling, and try to put this into their models to figure out what this means for Arctic food webs and Arctic seal populations. And I'm just putting this image up here because these are all the guys that go out and collect the samples with the subsistent hunters, and this work's not possible with them. So we have, without them, Martin and Tora down there on the bottom from Norway, and then David, Steve and Gary, who are based in Canada. So some subliminal messaging, if you like. What can we do to help reverse climate change? There's this really fantastic website called Drawdown, which is looking at solutions to try and reverse climate change. And a lot of those solutions are actually at a governmental level. They're not things that perhaps you or I could do, but the top two are things that we can do. And the first is to start eating a more plant-based diet, so I might become a vegetarian again. And the second is to start thinking about our food waste. This image is actually on their website, and it's from a farm up near Southport, and this is all food that's been wasted because it's the wrong size or shape for the supermarkets. So thinking more about where you get your food from is certainly going to help. And the other thing, of course, is thinking about your transportation as well. So I'll just leave you with that thought to end the talk. Thank you very much for listening.
Thank you.